Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In this video, we're going to be discussing the chromosomal basis of inheritance. When Mendel's hereditary factors were first proposed, these ideas were purely abstract, and many scientists were very skeptical as to how this can work. However, as the processes of mitosis and meiosis were worked out, biologists began to see parallels between the behavior of Mendel's proposed hereditary factors and chromosomes. Around 1902, Sutton and Boveri and others independently noted the parallels, and the chromosome theory of inheritance began to form. The first solid evidence associating a specific gene with a specific chromosome came in the early 20th century from the work of Thomas Hunt Morgan. These early experiments provided convincing evidence that the chromosomes are the location of Mendel's heritable factors. So we're starting to be able to put pieces of the puzzle together, seeing that these genes don't just randomly float, that they are associated with chromosomes, and then chromosomes are going to be the location of Mendel's heritable factors. Morgan was also studying genetics, but instead of pea plants like Mendel chose to study, Morgan studied Drosophila melanogaster, which is a common species of fruit fly. There are several characteristics that make fruit flies a convenient organism for genetic studies. The first is they produce many offspring. A generation can be bred every 2 weeks, and they have only 4 pairs of chromosomes. So all of these things make fruit flies a very convenient organism for genetic studies. In his studies, Morgan noted wild type, and the wild type is the normal phenotype that was common in the fly populations. So what was seen normally, those phenotypes were referred to as wild types. Any traits that were alternative to the wild type are called mutant phenotypes. The first mutant that Morgan discovered was a fly with white eyes rather than the wild type red eyes. In one experiment, Morgan mated male flies with white eyes, which are the mutant type, with female flies with red eyes, which were the wild type. The F1 generation all had red eyes. The F2 generation, when he crossed the F1 generation together to make the F2 generation showed a 3-to-1 red to white eye ratio, but only the males had white eyes. Morgan then determined that the white-eyed mutant allele must be located on the X chromosome. Just like in humans, female flies have 2 X chromosomes, while male flies have 1 X and 1 Y. Morgan's findings supported the chromosome theory of inheritance, that offspring inherit genes through chromosomes. Morgan's work not only showed that a specific gene is carried on a specific chromosome, but his work also showed that genes located on sex chromosomes exhibit unique inheritance patterns. A sex-linked trait is a trait that is determined by a gene that is on the X chromosome. This is different than those genes that are on autosomes, which are the 22 other pairs of chromosomes. When we were talking about Mendelian genetics, we were talking about inheritance through autosomal chromosomes. However, when we talk about sex-linked traits, we are talking about traits that are determined by a gene on the X chromosome. As we just mentioned, females are XX and males are XY. Those Y chromosomes of the male are condensed. There are few genes that are found on the Y chromosome, few genes that are going to be expressed on that Y chromosome, and those genes are really just to give the male traits to a male. The recessive alleles that are found on the male single X chromosome, therefore, have no active counterpart. So if a male gets a sex-linked trait on the X chromosome, it is going to be seen or expressed. X chromosomes have genes for many characters that are unrelated to sex, whereas most Y-linked genes are going to be related to sex determination. There are short segments at the ends of the Y chromosomes, and these segments are homologous with the X chromosome. This allows the Y chromosome to line up with the X, behaving like homologous chromosomes during meiosis in males. So again, remember that the X chromosome has a lot more genes on it. It is bigger. The Y chromosome is smaller with very few genes. Those genes are going to be related to sex determination. But because of these short segments at the ends of the Y chromosome, it allows the X and the Y chromosomes to line up and behave like homologous chromosomes during meiosis for separation of those sex chromosomes. A gene on the Y chromosome called SRY, which stands for sex determining region on the Y, is responsible for the development of the testes in an embryo. Now, females have two X chromosomes, and as I mentioned already, those X chromosomes have a lot of genes on them, so females do not actually need both of these X chromosomes to be expressed in their cells. So one of the X chromosomes in females is going to be inactivated early in embryonic development. This is shortly after the embryo's sex is determined. Which X chromosome is inactivated in the female varies randomly from cell to cell. So the same X chromosome is not going to be turned off in every cell. It is random which one is inactivated. If a woman is heterozygous for a sex-linked trait, meaning she has one X that has the trait and one X that doesn't, then some of her cells will express one allele, and some of the other cells will express the other allele because it is random as to which X chromosome is inactivated. The inactivated X chromosome is highly condensed, making it visible as an intensely staining bar body. What it is called at that point is a bar body, and that is just a very highly condensed X chromosome, and it is inactivated and not working. So in every cell, the female will have one X chromosome and then one bar body that was the X chromosome, but that one wasn't activated or condensed. And again, it is very random as to which X is the one that is going to be condensed in the cell. Because of this random inactivation of the X chromosomes in the cells, females that are heterozygous for X chromosome alleles are genetic mosaics. Their individual cells may express different alleles depending on which chromosome is inactivated. An example that we can see physically are with calico cats. Calico cats are normally female, and they have dark fur, orange fur, and white fur, allowing them to be that calico look that we know. The dark fur is actually inactive where the orange fur is, and the orange fur is inactive where the dark fur is. And so these are sex-linked traits on the X chromosome, and depending on which X is turned off where, we can see that variation in either dark fur being expressed or orange fur being expressed. The white fur itself has to do with has to deal with pleiotropy, okay, which is the multiple alleles that are present. And the white fur actually comes from the piebald gene, which is on a different chromosome. A gene that is located on either sex chromosome is then referred to as a sex-linked gene. Genes on the Y chromosome are called Y-linked genes. There are very, very few of these types of genes that are linked to the Y chromosome. Genes on the X chromosome are referred to as X-linked genes. X-linked genes follow specific patterns of inheritance. For a recessive X-linked trait to be expressed, a female is going to need 2 copies of the allele, so the female would have to be homozygous for that allele, and a male only needs 1 copy of the allele, so he is referred to as hemizygous. So in order for these X-linked genes to be shown again in the female, a female would need 2 copies of the allele, so both X's would have to have the copies of the allele, whereas the male only needs 1 copy of the allele in the X chromosome. Because the males only need 1 copy of the sex-linked trait, X-linked recessive disorders are actually much more common in males than in females. Some disorders that are caused by recessive alleles on the X chromosome in humans include color blindness, Duchenne's muscular dystrophy, and hemophilia. Linked genes tend to be inherited together because they are located near each other on the same chromosome. Each chromosome has hundreds or thousands of genes, except the Y chromosome. Genes located on the same chromosome that tend to be inherited together are called linked genes. Let's take a look at how linkage affects inheritance. Morgan did other experiments with fruit flies to see how the linkage affects inheritance of the 2 characters. Morgan crossed flies that differed in traits of body color and wing size, and he found that body color and wing size are usually inherited together in specific combinations, which were parental phenotypes. From his experiments, he noted that these genes do not assort independently and reasoned that they were on the same chromosome. However, non-parental phenotypes were also produced. Understanding this result involves exploring genetic recombination, which is the production of offspring with combinations of traits differing from either parent. Offspring that have a phenotype matching one of the parental phenotypes are called parental types. Offspring with non-parental phenotypes or new combinations of traits are called recombinant types or recombinants. A 50% frequency of recombination is observed for any 2 genes on different chromosomes. Morgan discovered that genes can be linked, but that the linkage was incomplete because some recombinant phenotypes were observed. He proposed that some process must occasionally break the physical connection between genes on the same chromosome. That mechanism was the crossing over of homologous chromosomes, and we see the crossing over occur during meiosis 1 in the production of gametes, eggs and sperm. Because of this crossing over, recombinant chromosomes, chromosomes that have been recombined, bring alleles together in new combinations in the gametes. And then what happens after that is that random fertilization which is the random combining of the random egg with the random sperm, increases even further the number of variant combinations that can be produced. This abundance of genetic variation is the raw material upon which natural selection works. The next logical direction for these sets of experiments was to map the distance between genes using recombination data. Alfred Sturtevant, one of Morgan's students, constructed a genetic map. This genetic map is an ordered list of genetic loci along a particular chromosome. Sturtevant predicted that the farther apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher recombination frequency. In other words, the greater the distance between two genes, the more points there are between them where crossing over can occur. A linkage map is a genetic map of a chromosome based on recombination frequencies. Distances between genes can be expressed as map units, where one map unit represents a 1% recombination frequency. Map units indicate the relative distance and order and not precise locations of genes. A linkage map is going to be based on the probability of crossing over, and it's not showing exactly where the genes are, but based on their recombination frequencies is where they're going to be placed. So one map unit would equal a 1% recombination frequency, and so here we're seeing a 9% recombination frequency. So these genes are located 9 map units away from each other, and this other set would be located 9.5 map units from each other. So it's based on the data that is collected and how often recombination frequencies were seen. Genes that are far apart on the same chromosome can have a recombination frequency near 50%. So what we're seeing is what Sturtevant predicted was that the further the genes are from one another, the higher the recombination frequency. Such genes are physically linked, but genetically unlinked, and behave as if they are found on different chromosomes. Sturtevant used recombination frequencies to make these linkage maps of fruit fly genes. He and his colleagues found that the genes clustered into 4 groups of linked genes called linkage groups. The linkage maps that he created, combined with the fact that there are 4 chromosomes in Drosophila, provided additional evidence that genes are indeed located on chromosomes. The figure in front of you shows an example of a genetic map, a genetic linkage map. So this is actually a partial map that shows 7 of these genes that we can see in Drosophila. On the top, we can see the mutant version of the gene, and then on the bottom, we see the wild type or normal version of the gene. And this is a gene map, a linkage map, and what it shows is the map unit length distance from one gene to the other. Alterations of chromosome number or structure can cause some genetic disorders. Large-scale chromosomal alterations in humans and other mammals often lead to spontaneous abortions, also known as miscarriages, or cause a variety of developmental disorders. Plants can actually tolerate such genetic changes better than animals do. In a process called non-disjunction, pairs of homologous chromosomes do not separate normally during meiosis. As a result, one gamete receives 2 of the same type of chromosome, and another gamete receives no copy. Aneuploidy results from the fertilization of gametes in which non-disjunction occurred. Offspring with this condition have an abnormal number of a particular chromosome. Non-disjunction, as I just mentioned, is a process in which pairs of homologous chromosomes do not separate normally during meiosis. Now, we can see this not only in homologous chromosomes, but we can see non-disjunction happening with sister chromatids as well. Let's take a look at this figure in front of you. In meiosis 1, normally what we have is the separation of homologous chromosomes. If they do not separate correctly, then non-disjunction occurs. So in this example here in meiosis 1, this pair of homologous chromosomes did not separate correctly, and therefore they stayed together, giving one of the cells many more chromosomes than the other. What happens is now they split, and the gametes that form some have many more chromosomes than they should, and some have less chromosomes than they should. So this would be an example of non-disjunction occurring in meiosis 1, which means there is not that separation of homologous chromosomes that needed to happen. We can also see non-disjunction with sister chromatids. Sister chromatids separate in meiosis 2. So in this example, meiosis 1 went according to plan, but now what we're seeing is non-disjunction occurring in meiosis 2. And what's happening here is that the sister chromatids are not separating in this specific cell, and so it's leading to gametes that have more chromosomes than the other. Now, when non-disjunction occurs during meiosis 2 for the sister chromatids, we do have the possibility of getting normal gametes that we see here, and then a regular number of gametes in the eggs or sperm that were produced by non-disjunction. If a zygote, which is formed by the fertilization of the egg and the sperm, if a zygote has only one copy of a particular chromosome, it is known as monosomic. If that zygote has 3 copies of a particular chromosome, it is known as a trisomic zygote. Polyploidy is a condition in which an organism has 2 complete sets of chromosomes. We see this with 2 or more sets of chromosomes. So we can see this with triploidy, where it has 3 sets of chromosomes, and tetraploidy, which has 4 sets of chromosomes. Polyploidy is common in plants, but not in animals, and polyploids are more normal in appearance than aneuploids. There are some alterations of chromosome structure that we occasionally see. Breakage of a chromosome can lead to 4 types of changes in chromosome structure. Deletion removes a chromosomal segment. Duplication is when we have a repetition of a segment. Inversion reverses the orientation of a segment within a chromosome. And then the other thing that we can have is translocation, which moves a segment from one chromosome to another. Alterations of chromosome number and structure are associated with some serious disorders. Some types of aneuploidy appear to upset the genetic balance less than others. This results in individuals who can survive to birth and beyond. These surviving individuals have a set of symptoms or syndrome that is characteristic of the type of aneuploidy. Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21, so it is also known as trisomy 21. It affects about 1 out of every 830 children born in the United States. The frequency of Down syndrome increases with the age of the mother, and this is a correlation that has not yet been explained. Non-disjunction of sex chromosomes can lead to a variety of aneuploid conditions. Triple X females are healthy with no unusual physical characteristics. Klinefelter syndrome is the result of an extra chromosome in a male producing XXY individuals. Monosomy X, called Turner syndrome, produces females that have one X and no other sex chromosomes. So they just have one X, so they're referred to as XO females. These females are sterile. It is the only known viable monosomy that is found in humans. Disorders can also be caused by structurally altered chromosomes. The syndrome, Cri-du-chat, or cry of the cat, results from a specific deletion in chromosome 5. A child born with this syndrome is severely intellectually disabled and has a cat-like cry. Individuals usually die in infancy or early childhood. Certain cancers, including chronic myelogenous leukemia, abbreviated CML, are caused by translocations of chromosomes. We can also talk about the inheritance of organelle genes. Extranuclear genes, or cytoplasmic genes, are found in organelles in the cytoplasm. So these are not genes from the DNA in the nucleus, but genes that are found within organelles. Mitochondria, chloroplast, and other plant plastids carry small circular DNA molecules, and therefore they have genes of their own. These extranuclear genes are inherited maternally because the zygote cytoplasm comes from the egg. The first evidence of these extranuclear genes came from studies on the inheritance of yellow or white patches on leaves of an otherwise green plant. Some defects in mitochondrial genes prevent cells from making enough ATP and result in diseases that affect the muscular and nervous systems. For example, mitochondrial myopathy and Leber's hereditary optic neuropathy are examples of these diseases where there are defects in those mitochondrial genes.